So in this video, I'm going to go through the bacterial unknown PowerPoint with you guys. I'm just going to explain the project and how we're going to complete it and how to make a flow chart. At first, I'm just going to go through the example in the PowerPoint with you guys about the cats just as a warm-up exercise. And then we're going to go through the example at the end of the PowerPoint, which is also the example that you'll be completing for the quiz online. So as we begin, I just want to kind of remind you guys about what this project is about. It's about that game, or kind of like that game Guess Who, where you have to choose your opponent's, or you guess your opponent's card with you remember the game with the little faces that pop up. And so you want to ask as many questions as you can to eliminate as many of those people with one question as you possibly can. And so, for instance, if your opponent had a male, and you would want to ask, is your opponent female? And these questions can only be yes or no questions. And so they would say, no, my card is not a female. So you would push down all your females and you'd be only left with the males. So these are the kind of questions that you're going to be asking about your bacteria in order to identify your unknown sample. So the first example, and I'm going to put the screen up because I need the board to write on. I know it's hard to see the names, but the first example in the PowerPoint is these kitties. And in the kitty example, one kitty has gotten locked in your grandma's car, and she's called you in order to come and get the cat out. But in order to know which cat it is that you need, you have to ask your grandma questions about what the cat looks like. And grandma can only answer yes or no questions. So you need to look at all of the cats and think of an identifying characteristic that's a lot of the cats share in order to eliminate the cats. So, and we're going to make a flow chart to go along with the cats so that you can kind of get an idea of what the bacteria flow chart is going to look like. So, think about what the cats look like. One question that might eliminate a lot of cats at the same time is, does the cat have orange fur? So to make our flow chart, we're going to start with our question of orange fur, yes or no. So you ask your grandma, does your cat have orange fur? And grandma says, no, the cat does not have orange fur. So you get to mark out Fred, Tiger, Peaches, Teddy, and Maxine. We're just going to assume that Tiger is orange, even though he's this mysterious gray and grayer cat. He's a black and white photo. But he looks orange to me. So we're only left with Rocky and Sadie. So you're always going to write down on your flow chart the remaining unknowns that are left over to choose from. So now you look at Rocky and Sadie and you say, what is a characteristic that would help you identify Rocky over Sadie or Sadie over Rocky? So you can ask maybe your grandma, does the cat have white fur? And grandma says, yes, the cat does have white fur. So you can eliminate Rocky. And cartoon cat Sadie is the cat that's hanging out in grandma's car. So that is how the flow chart works with the cats. Now I'd like to go through the example at the end of the PowerPoint with the bacteria. And we can practice this together. So the first thing that you're going to need is the taxonomic reference guide. When I give you guys your, and there, I think there's a copy of this in Blackboard, or maybe I'm not sure, or Moodle, whatever you guys use. But it looks similar to this. So in the taxonomic reference guide, the bacteria are grouped in major groups. So each group has a number, and on your sheet, the number will have a description out here on the line. And this line here means, like, so if it says that they're all coccus, or they're all aerobic, or they're all catalase positive, this means that every bacteria in this group has that description. So you can use those descriptions in order to eliminate entire groups very, very quickly. So then once you've started eliminating groups, you can start looking at the bacteria individually and look at their test results in order to eliminate them as well. And so these are just the genus. We're gonna talk about the species in just a moment. So this is very similar to what your data sheet will look like for your unknown project. So you'll get this set of colored pictures, and each of these has a result for these tests that fits your bacteria. So you're gonna, first of all, look at these results, and you are going to say which which question could I ask in order to eliminate as many bacteria as I possibly can. Well, it stands to reason, since Graham stain divides bacteria into two different categories, then we might want to begin with it. So if we start with the Graham stain, remember, we can only be gram negative or gram positive. Our gram result here is positive. And also the gram can tell us about the morphology of the bacteria. You can see here the bacteria are coccus shaped. And so you don't need a separate test for that. You can just use the gram stain to determine the shape of the bacteria. So now we're going to flip through the taxonomic reference guide. You're going to eliminate every group that is gram negative. And then of the gram positives, you're going to eliminate all of the groups that are bacillus shaped. And it's only going to leave you with groups 17 and 19. And so because 19 says it could be bacillus or coccus, you're actually going to look at those two bacteria, the bacillus and the clostridium, and look and see both of those are bacillus shaped. So we can actually eliminate the groups 19 as well. And so we're only left now with group 17. So group 17 is only got 5 or 6 bacteria in it. So we've used the gram stain. Now, because we've eliminated everything within the group, you guys are going to look at one of these tests that all of those bacteria listed in that group have a result for. And so an example might be the catalase test here. So our next test will be the catalase. The test result here for catalase is positive. The catalase test is going to eliminate all of the bacteria on the list. The only catalase positive bacteria are Micrococcus and Staphylococcus. And so now we need a test that's going to help us differentiate between Micrococcus and Staphylococcus. In your sheet, it gives you their respiration differences. Says that Staphylococcus is facultative anaerobe, while it says Micrococcus is an obligate aerobe. So you look up here to your test results, and which one of these test results can you think of that would help you determine the metabolism of your bacteria? Well, I see the thioglycolate broth here, and so that's the next test I'm going to choose. And I'm actually going to move up here because I'm running out of room. So our next test is the thioglycolate. Thioglycolate has 3 different things it can tell us. It can be aerobic, anaerobic, or facultative. Our result says facultative. We look at the sheet, it tells us Staphylococcus is facultative. And so now we're left with just the genus Staphylococcus. Now, this is fine. Now, the next thing that you would do is move from the group listings with all the genera to the species list. So once you have one genus, you're going to look to the species list. So you'll look to species list, you will see group 17. Under group 17, you'll see that there are 2 species of Staphylococcus. There's Staphylococcus epidermidis and there's Staphylococcus aureus. And so now we need to narrow it down to just one. So we did some tests in class that actually help us determine the differences between Staph epi and Staph aureus. One of those is the coagulase test. So let's look at our coagulase. The coagulase result here is positive. You see how it's clotted at the end and it's not running down the side of the tube. We talked about in class how only Staph aureus has this result over Staph epidermidis. So now Staphylococcus aureus is our unknown bacteria. So please practice making this flow chart, and please practice this before you begin your quiz, as it'll be very helpful for the last 2 questions. Thank you.